the DNA. DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotide. A pair of nucleotide is referred to as base pairs present in DNA. Base pairs or number of nucleotides is also characteristic of an organism. For example, a bacteriophage known as 5174 has 5386 nucleotides. Bacteriophage lambda has 48502 base pairs. Escherichia coli has 4.6 into 10 raised to 6 base pairs. And haploid content of human DNA is 3.3 into 10 raised to 9 base pairs. Let us discuss the structure of such a long polymer. Structure of polynucleotide chain. Let us recapitulate, means summarize the chemical structure of polynucleotide chain, DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA, and deoxyribose in case of DNA, and a phosphate group. Remember that nitrogenous base plus pentose sugar is nucleoside, and nitrogenous base plus pentose sugar plus phosphate group is nucleotide. So when you add phosphate group to nucleoside, it becomes nucleotide. Cytosine is common for both DNA and RNA. Thymine is present in DNA. Uracil is present in RNA at place of thymine. So thymine is not present in RNA and uracil is not present in DNA. Nitrogenous bases are of two types: purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, uracil, and thymine. The trick to remember pyrimidines is CUT, cut. Okay, so for pyrimidines, remember cut. Nitrogenous base is linked to the OH of first carbon of pentose sugar through a N-glycosidic linkage to form a nucleoside. So here you can see this is sugar shown in purple color. Now this is nitrogenous base. This nitrogenous base is attached to this sugar at the first carbon. You can see here it is attached to the first carbon and it forms N-glycosidic bond. N here is because it is bonded with nitrogenous base. So this portion, sugar and base together, is called nucleoside. And when phosphate group is attached to this nucleoside, it is called nucleotide. Examples of nucleosides they have given: adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, uridine or deoxythymidine. This is important. Uridine or deoxythymidine. These are all nucleosides. Adenine is purine, adenosine is nucleoside, and adenylic acid is nucleotide. So here you can see adenine is nitrogenous base, adenosine is nucleoside, and here AMP means adenosine monophosphate. So these are nucleotides. Diphosphates, triphosphates, all are nucleotides. Guanine is nitrogenous base, guanosine is nucleoside, and GTP, let's say, is nucleotide. Cytosine is nitrogenous base, cytidine is nucleoside, and cytidine triphosphate is a nucleotide. Similarly, it is same for thymine and uracil. When phosphate group is linked to OH of fifth carbon of nucleoside through a phosphoester linkage, a corresponding nucleotide is formed or deoxynucleotide is formed depending upon the type of sugar present. So phosphate group attaches to fifth carbon of the nucleoside through phosphoester linkage. So here you can see this is phosphoester linkage. Okay, so this marked one. Okay, here you can see fifth carbon also. Fifth carbon. So this is phosphoester linkage. Two nucleotides are linked through 3-5- phosphodiester linkage to form a dinucleotide. OH at third carbon of first nucleotide reacts with phosphate, which is attached to the fifth carbon of the second nucleotide, and water is released and ester bond is formed. Here you can see there is a ester bond, phosphodiester bond. This is not given in the textbook, but this line is given, but this diagram is not given. I have added it to explain. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has a free phosphate moiety at 5' end. Here you can see at the 5' end there is free phosphate. At the 5' end of sugar, which is referred to as 5' end of polynucleotide chain. Similarly, at the other end of polymer, the sugar has free OH group at 3' carbon or at third carbon, which is referred to as 3' end of polynucleotide chain. 3' or 3' end of the polynucleotide chain. So here you can see there is free OH group at 3' end of this chain. The backbone of a polynucleotide chain is formed due to sugar and phosphate. Okay, so here you can see this brown color are sugars and phosphate. So these sugar and phosphates, they form the backbone of this polynucleotide chain. The nitrogenous bases are linked to sugar moiety. Moiety means part. Sugar moiety, and these bases project from the backbone. You can see this adenine, thymine. In RNA, every nucleotide residue has an additional OH group present at second position in ribose. At second position of ribose, there is OH group, and at second position of deoxyribose, it has only H. There is OH. The structures of ribose and deoxyribose are almost same, except at this second position, where in ribose it has OH in second position and in deoxyribose it has H. Also in RNA, uracil is found at place of thymine. 5-methyl-uracil is another chemical name for thymine. 5-methyl-uracil is thymine. DNA as an acidic substance present in nucleus was first identified by Friedrich Mischer in 1869, and he named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation, that is explanation of structure of DNA, remained elusive, that is difficult to find for a very long period of time. It was only in 1953, remember the date, 1953, that James Watson and Francis Crick, based on X-ray diffraction data produced by Morris Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. So the famous double helix model that you see of DNA today was proposed by James Watson and Francis Crick. So it is called as Watson and Crick model of DNA. One of the hallmarks of their proposition was base pairing between two strands of polynucleotide chains. The hallmark was base pairing between the two strands of polynucleotide chain. However, this proposition was also based on observation of Erwin Chargaff that for a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equals 1. Let's see that. So, according to Chargaff, for a double-stranded DNA, for a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine, ratio between adenine and thymine, guanine and cytosine are constant and equals to 1. So this is Chargaff's rule. Now this rule you can also remember it as A is equal to T and C is equal to G. That's all for this video. The rest part of the DNA will be continued in the next video. Thanks for watching. Keep studying. Best of luck.